Hello, hi everyone. I would like to share with you this very nice website. It's called Wiley Interactive Animations, so you can find the link from here. I will type it on Putra Blast later on. So what you can see from here with this interactive animations from Wiley, there will be different topics, and you can look into the concept reviews, and there's some interactive animations, some structured tutorials, and there are also quizzes that you can try online. So let's have a look into one of the topics, which is glycolysis. Alright, so this is the interactive concept that they have, and there is a flow of where does the enzyme match up. For example, over here, there's different types of enzyme. Pyruvate kinase, triose phosphate isomerase, so analase kinase. So where does it fit in into the series of glycolysis step? Okay, let's have a look. Let's try pyruvate kinase. It's supposed to be here. Maybe this one. Maybe this one. Okay. Right. So does it fit in or not? Let's have a look. So this is some reactant's name. So it's actually a quiz. Yeah. So you can read yourself. So it's a very nice way to study. Let's have a look. What else do they have? Okay. Let's go to Cori cycle. So this is the Cori cycle. So it gives some explanation. So like for example in this case, skeletal muscles have few mitochondria, yet are capable of various activity. As a result, the small stores of oxygen are rapidly depleted, causing the muscle tissues to operate essentially anaerobically. So the Cori cycle is a pathway in carbohydrate metabolism that links this anaerobic glycolysis in muscle tissue to gluconeogenesis in the liver. So we see here, this is how the muscle contracts. Alright, at this point, you will find that the contraction will deplete the ATP storage in the muscle. Right, so they start using all the ATP quickly. So the next step is that the ATP from the muscle contraction is supplied by a glycolytic pathway. So what happens is that this is via anaerobic method, whereby they will generate 2 moles of ATP, and the lactate produced is secreted into the bloodstream. Okay, so these are the steps. So this is anaerobic respiration. So you see that it stays in the cytoplasm actually, whereby it will produce lactate as the end product. Right? So there's some questions here, so you can answer it. So what happens during the lactate secretion? What happens? This is the series of pathways that is involved. So this gluconeogenesis step using 2-lactate. Right? So how they produce the 2-lactate from the glucose from the 2-lactate products. So let's have a look at other. Let's have a look at this one. Metabolic process location. So where does all the activity takes place? This is more to metabolic integration. This will be nice in your last lecture. We'll be looking into this part. So the digestive state, what happens? Digested, secretes, like pancreas. That's quite cool. Right? So let's see what else. The other day we spoke about pyruvate dehydrogenase complex. Let's have a look at what they have over here. So this is the PDC. This is where your pyruvate is going to be converted into acetyl-CoA before it is being used by the Krebs cycle or citric acid cycle. So where does it localize? Location. So you can see here it is localized. Its activity is localized in the inner mitochondrial membrane. Right? So this is where all the activities takes place. So you have the pyruvate translocase. Okay, so here's composition we discussed the other day that is made up of 3 different components. It's a complex. So this is the reactions. The pyruvate will be converted to acetyl-CoA. So this step here involves the 3 series of enzymes inside the pyruvate dehydrogenase complex. Okay, these are the basically the other 5 coenzymes we discussed earlier in the lecture. Let's skip this and look at the mechanism. So it's quite very complicated, but I think let's see whether they have animations for this or not. Okay, so you see this is the TBP, the thymine pyrophosphate on E1. Okay, so this is the pyruvate dehydrogenase so complex, so there will be a nucleophilic attached. Alright, so it's converted, then you have the carbon ion, so you release CO2, then the active acetaldehyde will go to the nucleophile attached, and then they will move forward and remove another carbon ion. It will be the oxidized lipoamide, and from there they will take up coenzyme A, reduce lipoamide, then moves forward to reduce FADH, NADH. Okay, so how it is controlled, you can have a look into here, right? You can see it for yourself. Yeah, so there are a few topics that you can actually look into. This is the Krebs cycle. So the citric acid cycle, a little bit on the introduction. So you know the citric acid cycle basically will receive acetyl-CoA or some of the components which are biomolecules intermediate from others, biomolecules such as amino acids, apart from going through the PDC in the form of pyruvate. So amino acids here, glucose and fatty acids. So move forward, so you find the cycle, just an overview of it, so the mechanism, how the energy is being produced, how to regulate it as well. It will be interesting for you to view it yourself later on. Okay? Alright, so enjoy trying these interactive animations. Hope this will assist your studies.